Pharmacogenetics is the combination of the word pharmacology and genetics. Pharmacology in itself means the study of drugs, while genetics is the study of how traits are inherited. So pharmacogenetics is the study of the linkage between an individual's genotype with an individual's response to a foreign compound. Each person has a unique genetic makeup that differentiates on how they will respond to drugs. Pharmaceutical companies aim to produce the medicine that can give efficacy to 1/3 of the population in order to be approved by the Food and Drug Administration, or the FDA. But while some of the population react well to the drugs, others are not so lucky because either they don't react at all to the drugs or even worse, they have an adverse reaction towards the drugs. Adverse reactions may vary from headache, nausea, and vomiting to coma or even death. Because of this, physician must adjust the dose of the drug, not too high or too low, so that to avoid toxicity and increase its efficacy, because there are no standardized drugs that fits all. For humans, our genome is 99.9% identical with each other, but that resounding 0.01% makes all the difference that gives us our own uniqueness that differentiates us. We have different faces, different fingerprints, eyes, color, and hair color. It goes to show that even a tiny little variation in the DNA can cause major changes in a person. One of the main sources of variation in the DNA is single nucleotide polymorphism, or SNP. SNP is the result of mutation in which a single nucleotide is substituted by another nucleotide at a given position. SNP occurs once every 300 to 3000 base pairs. If one compares the genomes of two unrelated individuals, on an average it can be said that one SNP occurs in a sequence of 1000 base pairs. Thus, in the entire genome, the estimate of SNPs would be around 3.2 million in 3.2 billion nucleotides. SNPs are responsible for most of the medically important SNPs, or traits that are related to the predisposition of the individuals to certain diseases and differential drug response. With current technology, we are able to study and utilize SNPs, especially those that have a clinical significance, such as common SNPs that occur at a frequency of greater than 10%, or rare SNPs with strong selection components and a more marked effect on the phenotype, which is also equally important. If we can know the frequency of these common SNPs, we can utilize this knowledge to predict an individual's genetic makeup and how it reacts to a certain drug, thus making a drug response profile that is useful for identifying patients who are at high risk to experience adverse drug reactions or to determine the best therapeutic approach for a given patient. SNPs will play a major role in establishing a genotype related to drug response. The body's response towards drug is a highly complex process requiring the use of many proteins such as carrier proteins, transporters, metabolizing enzymes, and receptors. Polymorphism that occurs in our genes coding for these proteins will alter its activity and expression. Metabolism provides an important pathway for the process of limiting the drug. And sure enough, polymorphism can occur in the genes that are supposed to encode enzymes to metabolize the drugs more frequent than any other genes. Liver is the main organ for drug metabolism as it produces many of the drug metabolizing enzymes. All these enzymes have distinct phenotypes due to the genetic polymorphism. Mutations such as deletion, null, or inactivation can lead to a poor metabolism, while gene duplication can give rise to ultra-rapid drug metabolism. Liver metabolizing enzymes can be classified into two groups, which is phase 1 and phase 2 drug metabolizing enzymes. CYP2D6 comprises of 1 to 2% of the total liver drug metabolizing enzymes, but it is involved in the metabolism of more than 30% of drugs. Genetic variations including point mutations resulting in an early stop codon or amino acid substitution, microsatellite nucleotide repeats, and gene amplifications or deletions are the causes of differences in the enzymatic activity of CYP2D6, resulting in either complete deficiency to ultra-rapid metabolism. The CYP2D6 for hydroxylated phenotype, or the so-called Spartin-Debrisquin allele, is caused by homozygous inheritance of inactivating alleles. The frequency of this trait varies extensively among ethnic groups, ranging from 6% in Black Africans to 51% in Asians. Consequently, clinical responses to standard doses of drugs metabolized by CYP2D6, such as haloperidol or chlorpromazine, codeine, and antiarrhythmic, vary from increased risk of ADRs at recommended doses in poor metabolizers to therapeutic failure in ultra-rapid metabolizers. CYP3A4 is involved in the metabolism of more than 50% of all drugs prescribed to date. The inter-individual differences in the pharmacogenetics of this drug is thought to be related to variation in CYP3A4 enzyme activity. The variation may be caused by the age and disease-related differences, by drugs inducing or repressing the transcription or translation, or by the genetic polymorphism. The allelic frequency for the CYP3A4-1b allele, which contains an 8 to 90G substitution in the promoter region of CYP3A4, was found to be more than 54% in African Americans, whereas in American and European Caucasian it was reported to be 4 to 5%. This mutation was not found in Chinese and Japanese Americans. Phase 2 conjugation of drugs can occur alone or after phase 1 metabolism. In order to enhance drug excretion in urine or bile, phase 2 enzymes link larger polar parts to drugs molecule, thus rendering drugs water-soluble ready for excretion. Several of these proteins have an important role in deciding drug response, such as uridine diphosphate glucuronidinotransferase A11, or we call UGT1A1, or N-acetyltransferase 2, or NAT2, and thiopurine S-methyltransferase, or TMPT. Polymorphic variation of the MPT can reduce biotransformation of thiopurine drugs and can lead to potentially fatal hematopoietic toxicity in some patients. UGT1A1 detoxifies various lipophilic chemicals and endogenous substances, including bilirubin. This gene contains polymorphism in the promoter region, which alters the level of expression for the encoded enzyme, resulting in a wide variation of drug metabolism. The homozygous mutation in inactivated NAT2 results in a slow inactivated phenotype. This phenotype is responsible for increased dose-dependent toxicity due to the accumulation of drugs such as the hydrolysine-induced lupus and isoniazid-induced neuropathies and sulfonamide-induced hypersensitivity reactions in some ethnic groups. We need to establish a strong correlation of genotyping testing with therapeutic outcomes for the application of pharmacogenetics in clinics. The reason why the principles of pharmacogenetics is progressing slowly is because the studies for it require large amounts of data set, such as in large prospective studies that evaluate the impact of genetic variation on efficacy of drug therapy and testing the hypothesis for different drugs, as well as for different polymorphism. At present, phenotyping test methods are established which allow one to classify a patient as PM, which is poor metabolizer, or EM, extensive metabolizer, but this requires the administration of a specific marker drug or test drug, collection of urine, blood, or saliva for analysis of drug and metabolite concentrations. Moreover, these tests are time-consuming, expensive, and subject to drug-drug interactions or other influences. Genotyping tests have the advantage of having to be done only once in a lifetime and provides unequivocal genetic information. However, at times phenotype may deviate from a genotype for a simple reason that phenotype is a manifestation of an enzyme or protein which itself can be subjected to activation, inhibition, induction, or suppression. Phenotype is also influenced by other xenobiotics in the system. Prediction is that with better understanding of correlation between phenotype and genotype, the trend will develop for genotyping as a test of predictive of phenotype. Pharmacogenetics will help to tailor the drug dosage according to the individual's genotype. Imagine it is like an archery where the arrow always hits the bullseye. It will help in minimizing the adverse drug reactions and will reduce the cases of inefficacies. People will be safe from the unnecessary exposure of drugs and their relative side effects. In the future, individual will carry a genetic profile and physician will demand for such profile before prescribing the medication.